Section 8.1 is on human chromosomes and genes. So we're talking about genetics still, but specifically with people. So our objectives for today: define the human genome, describe human chromosomes and genes, and explain linkage and linkage maps. The human genome is all the DNA that makes up a person, and it's a lot. So if we're talking about A's, T's, G's, and C's, those are bases. And of course, they pair up where T's and A's go together, G's and C's go together. Well, if you have a T and an A together, that's called a base pair. If you have a G and a C together, that's a base pair. So how big is the human genome? 3 billion with a B base pairs. So you could say 3 billion pairs or 6 billion letters. The Human Genome Project started in 1990 to try to do just that, catalog the entire human genome, and ended in 2003, so 13 years to do. And that's just to get the letters. Now we need to figure out what do they do. So what kind of genes are in there, and what letters code for what genes. And we know that humans have at least thousands of genes, probably in the 20,000s of genes, but we don't know where everything is yet. What we do know is that we have in all of our cells except for our sex cells, 23 pairs of chromosomes, 23 from mom, 23 from dad, so a total of 46. Of those 23, 22 are called autosomes. They don't determine whether you're a boy or a girl. So the first pair, the second, third, fourth, fifth, sixth, seventh, eighth, ninth, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, and 22 are all considered autosomes. We have, for the most part, the same numbers 1 through 22, whether we're a boy or a girl. There's a lot of important information on there, but gender is not one of them. Gender comes down to the sex chromosomes, the 23rd pair. And usually they don't even have a 23 next to them. They're identified as either X's or Y's. If you have two X's, XX, that means you're female. And you only need one working X chromosome. So as long as you have an X, you can be a living, breathing person. So if you have two X's, you're female, and only one of them is on at a time. So some cells have one X on, some have the other. Males get XY. So you've got the same X chromosome, and then this tiny little thing called a Y. It's not nearly as big as an X chromosome. It doesn't have the same information as an X chromosome. So for guys, one X is on, just like females, one X is on, but guys have this other thing called the Y chromosome that makes them guys, determines that they are male. And I have usually on there because there is a gene on the Y chromosome that has to turn on for somebody to become male. If that does not turn on, they don't become male. They stay female. We essentially all start developing as a female. However, some of us have a Y chromosome that will turn on a certain gene on there called the SRY gene that will make us male. That's why guys have nipples, because they started as girls. As long as it gets turned on. Now, if that doesn't get turned on, there are such thing as such people as an XY female, where they have the Y chromosome, but it just never turned on. The gene that needed to be turned on to become male never did turn on. But as long as you have one of the X's, you can survive, living, breathing human. As far as genes, sort of ballpark guessing. We know it's around 20,000. How much? 20 to 22, 20 to 25, depends on which source you look at and how old it is. At one time, they thought it was thought we had 40,000 or more. No, in the 20,000 range, which is more than fruit flies or nematodes, but not nearly as much as rice or corn, which is weird to think that corn's got more genes than us, but it does by far. But of those letters, those base pairs, only 25% of those letters code for genes. The other 75% does stuff that we're not sure about. Maybe it does nothing. We really don't know. We just know that 25% of those 3 billion are going to code for those 20,000 some genes. But keep in mind that you can have different genes. Like we all have the gene for eye color, but we have different forms of that gene, different alleles. Some might have brown, some might have blue, some might have green, etc. Linkage is how we determine, or part of how we would determine, where these genes are. If genes tend to be expressed together, for instance, eye and hair color, dark brown eyes go with dark brown hair quite often. Lighter colored eyes go with lighter colored hair, and even skin color as well. Lighter colored skin with lighter colored hair, darker colored skin with darker colored hair. They tend to be together because they're on the same gene. So if they are on the same gene, then no matter what happens through mitosis, meiosis, you end up with gene 3 and gene 4 being expressed together because they're on the same chromosome. They move together. You get combinations that usually go together. If they were not linked, they'd be on different chromosomes. You get all kinds of different combinations. If gene 5 and gene 6 are on different ones, for example. Gene 5 and 6 from mom and gene 5 and 6 from dad. On different chromosomes, you're going to get a whole bunch of combinations. But if they're on the same chromosome, not so much. There are certain genes that are called sex-linked because they are only on the X or only on the Y. Usually they're on the X. The Y chromosome is such a tiny little chromosome that there's not a lot of genes on there. The X chromosome has around 2,000 genes. The Y has less than 100. And since the Y chromosome is not necessary to be alive, women survive just fine without one, there's not a lot of genes on there of great importance. Anyway, sex-linked genes lead to interesting patterns in how certain, oh my gosh, I froze, in certain characteristics and certain diseases. So color blindness, hemophilia are a couple examples I can think of right off the top of my head that have to do with being sex-linked. So let's go through what that's like. So let's say color vision. Normal allele is that you can see all the colors. A defective allele is that you are colorblind. It's maybe red-green colorblind, maybe more with the blues and the purples. There's different kinds of color blindness, but we'll say red-green colorblind for now. So mom has the normal allele for normal color vision and the defective allele for red-green color blindness. However, since she has a good one, a normal one, she is not colorblind. She can see everything just fine. However, she carries it. Let's say dad has normal color vision. He has on his X chromosome the normal allele. Now, he doesn't have anything on his Y, so whatever he gets from his X, essentially whatever he gets from his mother, is what he's got. because the Y chromosome doesn't have room on it for color vision. If you were to do a Punnett square, essentially you could have that pair with that, or that pair with that. This one pair with this one, or this one pair with that one. If it was these two pairing, you'd end up with this child. Two X's, so a female, and has a normal allele from mom and a normal allele from dad. She will have normal vision. The second option would be this X from mom and this X from dad. Two X's, so female, has the color normal gene, is carrying the color blindness gene, but is not colorblind. Because she has the normal gene, she's okay. Possibility would be Y from dad. Y's always come from dads. And this X from mom. Well, this boy would have normal vision because he has the normal allele from mom. Last boy, this Y from dad, got this X from mom. That is the defective one, the one that has red-green color blindness. He's stuck with whatever he gets from mom because the Y that dad gave him did not have another copy of a color vision gene. So oftentimes with things like color blindness, hemophilia, females tend to be carriers, males tend to be affected. However, if you had, let's say, completely different families, because that's just weird, but this X from this boy combining with this X from this girl, you could have an XX colorblind female. It's just a little less common. As far as determining not only what chromosome genes are on, but where, has to do with how close are they. There was a thing in meiosis called crossing over, where the chromosomes would come together and exchange chunks of DNA. Well, if chunks of DNA are really, really close together, chances are crossing over is not going to split them apart. So these duplicated, crossing over occurred, but they ended up together still. If these two duplicate and some crossing over happens down here, you might end up with some different combinations. The farther apart they are, the more likely they're going to get split up during crossing over. The closer they are, the less likely they're going to get split up during crossing over. So if we know that genes are together often enough that we know they're at least on the same chromosome, but they end up with different combinations quite often, they would be far apart. If we know that they're on the same chromosome and they almost always are in the same combinations, they must be really close together. So information like that's how we determine not only what chromosome genes are on, but where on that chromosome. And if you start to figure out where, you can put together what's called a gene map. Now this gene map is not for a human. There are 1, 2, 3, 4 chromosomes. It is for the fruit fly. Fruit flies are an animal that we know a lot about because they're small, they reproduce quickly, and they're easy to take care of. So they end up being great for genetics experimentation. But we know now, and so this would be our centromere for chromosome 1. These are all the genes that we know the location of them. So they're on chromosome number 1 on location 33.0 for vermilion eyes. Chromosome 2 has these genes in the fruit fly. Here's your centromere. Chromosome 3, there's a centromere. 4 just has a centromere with all the stuff below. 4 is a smaller one. There's some weird ones. Raisin eye, eyeless, sparkling eyes, oh, that's nice, and dumpy wings. That sounds bad. To summarize, human genome consists of about 3 billion base pairs or 6 billion individual pairs of DNA. 2003, the Human Genome Project finished sequencing all 3 billion. Humans, like you and me, have 23 pairs of chromosomes. Of these, 22 of those are autosomes. The X and Y are the sex chromosomes, the 23rd pair. Females have 2 X's, one that's turned on and one that is not, and males have 1 X and 1 Y. Our chromosomes, all 23 pair, have between probably 20 to 20,000 genes. Usually, those genes have 2 or more alleles, or 2 or more forms. Linked genes are on the same chromosome. If we talk about sex-linked genes, they're on a sex chromosome, X or Y, usually on the X, because the X is so much larger than the Y. And how often crossing over happens between genes helps us figure out where they are located. So if they're linked, they're on the same chromosome. If they cross over together quite often, they're close to each other on the same chromosome. And that's what we can use to construct linkage maps.